Previously we saw how controlled crosses coupled with mass selection can improve an existing crop or create a new variety that bears a unique combination of desired traits. These within-species crosses have proven their efficiency since they have first been used by farmers and breeders. However, they do have one important limitation. They can only work with the genetic variability available within a species. So the extent to which they can improve a crop depends on the available genetic variation of said species. Sometimes a breeder wishes to improve a crop with a trait that exists in a different species. In this case, classical within-species crosses are not enough, and between-species crosses have to be carried out. These are called wide crosses. More generally, wide crosses are hybridizations between species that are genetically distant, and sometimes can include two varieties of the same species that have differences in their chromosome numbers. Some species can naturally hybridize. For instance, plums and apricot flowers can be cross-pollinated to produce plum cuts. In Jamaica, there is a fruit named ugly, which is a natural hybrid of grapefruit and tangerines. These are hybrid plants that have occurred relatively recently. But in fact, modern genetic analyses have shown that many of the cultivated crops are descendants of an interspecies hybrid that was made a long time ago. Interspecies hybrid crops are so ubiquitous because they are the result of countless trials of natural and human-mediated hybridization over a long period of time. Wide crosses are nevertheless difficult to achieve because of the reproductive barriers between species. These barriers assure the species' genetic integrity. They can be prezygotic, preventing the occurrence of fertilization and hence the formation of a hybrid zygote, or postzygotic, preventing the development of a hybrid zygote. Prezygotic barriers of interspecies hybridization occur because the pollen and pistils are capable of recognizing each other thanks to signalization molecules on their surface. If a pollen from another species is deposited, there will be no recognition between the signalization molecules on the surface of the pollen and the receptors on the surface of the stigma. The stigma will remain non-receptive and the pollen could not germinate. This can be avoided with pollen mentoring. First, the stigma is activated by a small amount of compatible pollen from the same species. Once the stigma is receptive, any deposited pollen, including from other species, could germinate and proceed with fertilization. Another way to avoid prezygotic barriers is to use a three-part pollination system. The ovules from one species, which is the pollen receiver, are removed from the pistil and deposited in a nutritious environment. The pistil from the other species, the pollen donor, is also removed and deposited in the same environment. Then the pollen from the donor is deposited on the isolated pistil. The recognition between the pollen and the pistil will occur because they are from the same species. Then the pollen will germinate, producing its pollen tube, and because there is no reaction of recognition between the pollen tube and the ovule, there will be fertilization between the two species. Finally, the whole pollination process can be circumvented altogether, and interspecific hybrids can be produced by directly fusing the cells of two different species. The cells are first chemically or electrically treated so that their membranes are more fragile. Then the two different cells come in contact. Their cytoplasm will fuse, producing a heterokaryon cell or a cell with two different heterogeneous nuclei. This is followed by the fusion of the nuclei, resulting in a chimeric cell or a hybrid that contains the chromosomes of two different species. Some types of rye have been produced with this protoplast fusion technique. In some cases, it is not the actual production of the seed embryo that is problematic. You likely know that plants undergo a double fertilization, one for the production of the embryo and another one for the production of the endosperm, which is the nutritious tissue surrounding the embryo in the seeds. Without this endosperm, even if the embryo is viable, it will quickly die after germination because it will have nothing to feed on. Sometimes the problem with interspecies hybridization is not the viability of the embryo, but the production of the endosperm, as is the case in these wheat seeds, which all have variable embryos of the same size, but their endosperm is highly underdeveloped. The lack of endosperm can be easily solved by embryo rescue. The embryo is excised from the seed and then deposited on a nutritious environment that will provide all the nutrients it needs during the germination and until the embryo or the seedling is capable of producing its own food via photosynthesis. But the problems of white crosses do not always stop after a successful fertilization. Quite often, the interspecies hybrids are sterile. They are not capable of reproducing themselves. This is because of chromosomal incompatibilities between the two parental species. An interspecies hybrid that has inherited two sets of chromosomes with different chromosome numbers will encounter problems during meiosis. Specifically, during metaphase, the separation of the two sets of chromosomes could not be achieved if the cell detects incorrect chromosome alignment due to differences in the size, structure, or number of chromosomes attached on both sides of the meiotic spindle. But even if the division does proceed, the resulting gametes will be non-viable. They will have two incomplete or aneuploid sets of chromosomes, so they will be likely missing some essential genes for the plant development. Such problems with chromosome numbers during meiosis are obviously not desirable if you want to reproduce the hybrids. One exception to this is the production of seedless fruit, which is purposefully made sterile via hybridization. If seedless fruits are not the goal of the white cross, we can produce fertile interspecies hybrids through polyploidisation. The chromosomes of a hybrid cell can be duplicated with some chemical treatments so that there are two copies of each chromosome set. Instead of a diploid, we will now have a tetraploid cell. When meiosis occurs, the doubled chromosomes will be perfectly aligned and paired with each other during metaphase, and the cell division will proceed normally. The result will be viable gametes that contain one set of chromosomes from each of the two original species. Many of the crops we use today are actually polyploids. Wheat is triploid, meaning it has 3 sets of chromosomes, potatoes are tetraploid, and strawberries can be anything from diploid to non-haploid depending on the variety. Let's see how interspecific hybridization was used to produce a hybrid plant destined for biodiesel production. Jatropha carcas, commonly known as the Barbados nut, is a wild perennial bush growing in the tropical parts of the American continent. Its fruits are rich in oils that can be used for biodiesel. Since this species has not been domesticated, it shows asynchronous flowering and variable yield. It also has 22 chromosomes. A recent breeding program aimed at improving the agronomic traits of the Barbados nut by hybridizing it with Ricinus communis, or the castor oil plant. This species has been domesticated long time ago, and although its fruits are toxic for human consumption, as most people learned through an episode of the cult series Breaking Bad, castor oil is often used as laxatives or in cosmetics. A wide hybrid between these two plants might have just the right properties required for the industrial production of Barbados nut oil. So first, the two species were crossed using controlled pollination. Then the F1 embryos had to be rescued and grow in a nutritious environment to obtain adult viable F1 plants. Then, because the number of chromosomes in the two parent species is not the same, the F1 hybrids, or more specifically their germ cells, needed to undergo polyploidization in order to be able to produce viable seeds. The breeding program is still in progress, and the polyploid interspecies hybrids are now undergoing controlled crosses followed by mass selection to eliminate all undesired traits. But already the F2 polyploid hybrids show traits of interest such as synchronous flowering and high yield.